Hi, my name is Bridget Keenan. I'm here from University of California, San Francisco, and I'm here to talk about my CCF Foundation Award project, which is the role of cancer-associated myeloid cells in resistance to immunotherapy in biliary cancer. By way of disclosures, we've received research funding from Partner Therapeutics, and a clinical trial that I'll reference was funded in part by Merck. Today I'm going to talk about biliary tract cancers. That's what we're all here at this conference to talk about, and how checkpoint inhibitors, as you heard, have not been quite a success in this cancer type thus far. In my project, I use multimodal sequencing to investigate mechanisms of resistance to immunotherapy, and in particular, I focus on monocytes, a type of myeloid cell. And finally, I'll come back to talk about how these monocytes may be related to T cells and in fact potentially suppress T cell responses. As somebody has already shown this slide before today at this conference, biliary tract cancers have proved particularly resistant to checkpoint inhibitors like Opdivo and Keytruda. This is data from the Keynote studies that show there are some patients that respond, whose tumor responds. However, the response rates typically are about 5 to 15% overall. And this is a disease that needs additional therapies. In this study, we looked at the blood of patients who had been treated with anti-PD-1 or pembrolizumab, a checkpoint inhibitor. We also had blood from healthy donors that we profiled in addition to the biliary tract cancer patient's blood. And we were able to look at serial time points at baseline and after treatment with anti-PD-1. The methods we used were something called site-seq or proteomics, where we're able to not only do single-cell RNA sequencing on the blood cells, but also use a panel of antibodies for about 100 different cell surface proteins. And this gives us information at a single-cell level of both the RNA and protein content of the cell. Here you're seeing what's called a UMAP. This is a plot that shows all of the different cells that are present in our data set, both from the healthy donors as well as the biliary tract cancer patients. It's a way to sort of simplify how these cells group based on their RNA expression profiles. And here, what I'm showing in the first main UMAP plot is we're able to identify all the major immune cell types from the blood of the healthy donors as well as biliary tract cancer patients. I then use this heat map to show an example of how these cells are differently represented between patients whose tumor responded and did not respond to anti-PD-1, as well as looking at the time points at baseline and after treatment with anti-PD-1. What we saw was that there were marked differences between the time point as well as response category. And one area where we saw that was particularly interesting was the myeloid cells. To focus further in on them, I grouped those cells together and clustered again using the single-cell RNA sequencing technique just on the myeloid cells. So this includes classical, non-classical monocytes, and dendritic cells. I'm going to focus, however, on just a few kinds of monocytes for this talk. What we found was that there were several different monocyte subpopulations, and we named these based on their RNA expression profiles and the different pathways that were overrepresented within these cells. The first one there in red at the bottom was overrepresented within healthy donors, and we called these CD14 IFL for inflammatory. These are cells that had an RNA expression profile that was consistent with normal, healthy immune function that would be expected of monocytes. We also found two populations that were predominant within cancer patients. The CD14 CTX cells, these were defined by what we called CTX or chemotaxis. So they expressed many different chemokines as well as proteins that are involved with breaking down the stroma and potentially involved with metastasis. And then we had another main population of monocytes that I'll talk about called the CD14 APCs for antigen processing. And they expressed a lot of different molecules that are involved in antigen processing and presentation. So also what would be expected from monocytes in terms of normal, healthy immune functioning. We are particularly interested in these CD14 CTX cells because what we saw was that in the blood of patients whose tumor did not respond in the time point 3 weeks after they received anti-PD-1, there was an increase in these cells. Whereas in patients whose tumor responded to anti-PD-1, we saw the opposite. We saw an increased frequency in the CD14 APC or antigen processing and presentation profile. What we saw when we compared just these two with differential expression testing was that the CD14 CTX cells upregulated a lot of different inflammatory molecules that I already talked about, like chemokines, but they also expressed inflammatory cytokines like IL-8 that can potentially be suppressive to the immune system. We also went back to the site-seek technique that I talked about, which we are able to look at proteins that are expressed on the cell surface. And we identified 2 of these as enriched in the CD14 CTX population. One is CD29, which is an integrin and involved in cell adhesion. And another one is TIM3, which is an immune checkpoint, which is typically thought about being expressed on T cells, but has also been shown to be expressed on different myeloid cell types like dendritic cells. We were then able to make a flow cytometry panel based on those markers, as well as CD14, a marker for monocytes, and show that this population was specifically enriched in biliary tract cancer patients, both in this cohort, validating our RNA and site-seek data, and also in a different cohort of biliary tract cancer patients. We then wondered, what is the functioning of these cells and how they might potentially be related to T cells, which are thought to drive anti-tumor responses. I don't have time to talk about all the different T cell populations today, but we did the same thing where we subsetted these T cells out from the main data set and focused in on them and looked at all the different expression profiles of these different T cell populations. And again, I won't describe all of these in this short talk. However, one thing we noticed was that these CD14 CTX potentially suppressive monocytes were correlated with a population of CD4 T cells that are expressing this marker called SOX3. This was interesting because SOX3 has been associated with what's known as an immune paralyzed phenotype, where T cells are not able to secrete cytokines and perform their normal immune function. We wondered whether this was a direct effect of the CD14 CTX cells. So we sorted on these cells using those markers I talked about in one of the previous slides, CD29 and TIM3. And we also took cells from healthy donors, healthy monocytes. We co-cultured those with CD4 T cells that were from healthy donors. And what we found was that the population of cells derived from the circulation of patients with biliary tract cancer were able to induce the SOX3 marker. However, healthy monocytes from either the same donor or a different donor were not able to induce the SOX3. We also looked at the secretion of cytokines by SOX3 positive and SOX3 negative T cells and found that SOX3 positive T cells did not secrete many cytokines like interferon gamma, TNF alpha, and IL-2 after being re-stimulated. So in conclusion, we found many different monocyte populations, and this is something we're able to do through this single-cell RNA sequencing technology that allows us to take a very deep look at immune cell populations in circulation. We found that there are several monocyte populations that were found preferentially within the circulation of biliary tract cancer patients compared to healthy donors. And one of these, the CD14 CTX population, had a suppressive chemokine and cytokine signature. This population was associated with resistance to anti-PD-1, particularly on treatment. And in addition, we found that these cells correlated with CD4 T cells that express a high level of SOX3, and these cells may be immune paralyzed from the results of our co-culture experiments. So there's a lot more work to be done here. I do have to thank the foundation first and foremost for allowing me to continue this work in the lab of Dr. Larry Fong with this award. I also want to thank Dr. Katie Kelly, who's here and has given several talks at this conference this year and as well as other years, for introducing me to the foundation, for providing the samples from this clinical trial that allowed me to do this work, and for being a fantastic mentor. And I also, in particular, want to thank all of the patients, both those who we've treated at UCSF and donated their samples to our studies, as well as to all the patients who are here. Thank you so much.